It is my great pleasure to present to you today the Revity Signals Notebook, an integrated cloud solution for research. I am Nimesh Joseph, an application scientist at Revity Signals, and a biologist by training who has extensively done wet lab work in the past. This makes me understand how valuable it is to digitize laboratory data, to make them findable, accessible, interoperable, and reusable, thereby conforming to the FAIR data principles. You might be at different stages of your digitization journey of your laboratory work. Currently, you might be capturing your data in paper notebook or in various different file formats. It could be Excel files, or it could be a GraphPad Prism file. If you have sequences, you might be using various different proprietary bioinformatics tools such as SnapGene or DNA Star. You might be looking to improve and digitize your process and thereby removing bottlenecks in terms of data visibility. What an ELN can do for you is to digitize and standardize your data capture. It can help you to manage and track your materials. It helps with data sharing and collaboration between colleagues. And most importantly, it helps you for your IP protection. Signals Notebook is very user-friendly. It allows you to easily search and report your data that you have captured in your electronic notebook. So the value that you get out of Signals ELN is everybody in your organization would make decisions based on a single source of truth. This gives you consistency, traceability, and visibility of your data. Thereby, you have less oversight and you have more insight into the data that you have captured in the ELN. Signals is a 100% cloud-based collaborative platform designed using the best available technologies so that your Signals Notebook is available anytime, anywhere, on multiple devices. This means you can access it using any web-capable device through any of the standard web browsers, Chrome being our recommended browser. Signals is built on modern microservices, which makes it extremely responsive, running parallel processes, providing fast, modern, and scalable performance. In terms of user experience, it is extremely intuitive. The system is incredibly fast. You can do Google-like search within the ELN. The notebook allows you to drag and drop files into your experiment. We have Microsoft Office integrated into the ELN so that you can create Microsoft Office documents from within the ELN, or you can drag and drop files and continue to edit them from within the ELN. The ELN is cross-platform compatible, which means you can work from Mac or Android devices. The Signals Notebook is built to make collaboration easy. The access control is designed for collaboration within the team and with external collaborators as well. You can send notifications to your colleagues from within the system. You can comment on experiments. There are signing and reviewing workflows built into the ELN. You can also create tasks for your colleagues, and you can request materials from within the ELN. Signals Notebook allows integration with other external systems, for example, your legacy ELN. We have a wide range of applications built into the Signals Notebook. One being ChemDraw, which is part of our product portfolio, which chemists can use for creating reactions, or it could also be used for creating new modalities in biologics. We have SnapGene integrated to handle sequence files. There is a Microsoft Office integration built into the electronic notebook so that you can create or edit Microsoft Office files from within the ELN. We have inventory application to track your assets. We also have partnership with other organizations, for example, Cytara for instrument integration. So the agenda for today's talk are as follows. We will look at how Signals Notebook can be accessed and how easy it is to navigate through the system. We will look at how experiments can be created and different types of data can be captured. We will look at asset management as part of the materials library. We will look at the experiment review and approval process. Data which is already captured within the ELN can be exported and shared with your colleagues and collaborators. We will also look at the audit trail and how we can track the history of an experiment. Finally, I will summarize my talk and we will have time for Q&A at the end. Now let us explore the Signals notebook in detail. I am logging into the system with my username and password and have reached the landing page. From here, I can decide what I would like to explore. Let me show you a couple of quick tips at the start of this demonstration. Under your name on the top right, there are some options for you to explore. It could be your user settings, or maybe you need some help with how to use the system. There are quick start guides available from the user interface. You also have links to what is new and recently released as part of the product. As a user, if you would like to have new capabilities incorporated into the product, you can contact our product team by using the Ideas portal. Let us go to the user settings. If you would like to upload a profile picture of yours, the system allows you to do that. This helps you to recognize entities and experiments that you have created, as you would be able to see your photograph associated with it. A key setting that users might be interested is the notification setting. You can toggle on and off to receive notifications within the app or through email. I'm going into Signals Notebook where I have reached Dashboard. The dashboard is highly configurable based on how you would like to look at the information that is available within the Signals Notebook. Here I have organized various different wizards that brings me the records that is already there in Signals Notebook. Whether it is all my recent experiments or my favorite experiments, I can see all my notebooks. Based on the review of my experiments, I can see those experiments which are waiting for my review, or my experiments which are under review, or any experiments which were rejected by my reviewer. We can also look at instruments which are coming up for calibration, or we can have a list of tasks that are assigned to me. Let us now look at some basic concepts in the Signals Notebook. On the top left, you see various different functionalities. First, the search options. As you move to the right, you can see the smart folders for all your notebooks or the experiments, or any requests or tasks that are created in the notebook. You can also explore materials library if that is what you would like to do. To create new experiments, you go to Add New and click on the experiment. This brings up a couple of options for you. If you would like to use a template, you can select the template from the drop-down list. Alternatively, you could create an experiment from a blank canvas and add the various different information elements that you would like to incorporate to capture the data as part of your experiment. A description is a human readable description of your experiment so that you can quickly identify what you have done in that particular experiment. The name of the experiment will be auto-generated by the system as configured by the system administrator. And there is flexibility in how you would like to create the name of the experiment. If you would like to assign this experiment to a notebook, the option is available to you. There are a couple of fields or attributes that the system administrator has configured and made it mandatory, as you can see with this asterisk against the fields. You could select the business area from a drop-down list, a department, and a project name if that is what your organization would like to capture for that particular experiment template. Let us now look at a biology experiment that I created using a template. The system administrator has created various different sections or pages for this particular template, as you can see on the left. The same pages can also be found as tabs at the bottom. If you would like to add a new page, you can use the plus icon here and rename that particular page by double-clicking on its title. There are various different options to capture data into your Signals notebook. Whether it's a text element, if you would like to create molecular structures, or in terms of more complex biologics, we can use the ChemDraw canvas. You can create Word, Excel, or PowerPoint files within the notebook. You can call in materials from the materials library. You can create samples. If you have plates associated with your experiment, you can create plate maps. If you have structured table to be used, you can call in tables which are configured in the system by the administrator. You can also create tasks, and you can also call in tasks that are assigned to you to be carried out. The text element will look very familiar to you. It is almost like a Word document where you can copy paste or type in, and using the various options to format the text. The drug target information comes from a library that is created in the materials library. If you'd like to add a new target, you can click on the search icon and select the target from the library. Signals Notebook will also allow you to make live calls to external databases. In this particular case, it's a UniProt. If you put an accession number for any particular protein that you are interested in, it will bring in information from UniProt. You can incorporate the materials used as part of this experiment as well. I have various different materials tables that I have incorporated. You can either search in the materials library by using the search icon. Alternatively, if you have a barcode, you can scan the barcode using a barcode scanner, which will populate the barcode into the ID column and will fetch container in which that particular reagent is stored. You can see all the information associated with material that is captured during the registration. In this particular case, that we can see vectors used for expression, the antibody chains, whether it's coding DNA sequence or the protein sequence. You can also see any documents which are associated with material that you are using. If you would like to see it, you can download and look at it in further detail. The materials library is communicating with the inventory, which means that any amount that you use as part of your experiment can be removed from the total amount that is available. In this particular case, I'm going to use 20 microliter of the antibody. And as you can see, the inventory is updated live as you have used 20 microliter in this particular experiment. Let us look at the materials library quickly. By clicking on this hyperlink or the ID of material, you will be immediately taken to the materials library. As you can see, the system can capture the parent level information, and all the fields that you see here are highly configurable by the administrator. Any batches of the material is captured under the material batch. You can also see the containers where material is stored, whether it's in a fridge or a freezer. Clicking on the container will take you to the inventory itself, where you can see the hierarchy of the inventory and the exact location where that particular material is kept. This is where you can see the total amount available and the barcode that is associated with that particular container. You can also go into further details by looking at the box level information, and clicking on any grid in the box will tell you exactly which material is kept in that particular location. As part of the materials used, I have the control antibody. I have used a couple of cell lines, and I can also add in the equipment that are used. If your business rule allows you to select standard operating procedures that are set up on the system and approved by a team leader or a group leader, you can call in such SOPs into your experiment. I am using SOP for antibody biophysical assays. All the information associated with the SOP will be brought into the SOP table, including the version, when it was reviewed, and who is the approver. If you would like to capture additional information about the methods that you used or any alterations that you did for the experiment, we can easily use a text element for that. Again, the system allows you to use text snippets, which are chunks of text where there are placeholders for you to click and select relevant information. These placeholders will allow you to select options from a drop-down list. If you have more complex experimental setup, we can have worksheets where individual steps can be clearly mentioned, and there can be tick boxes or timestamps that can be incorporated, as well as comment boxes where you would like to write up specific comments against that particular step. We can also have lookup into the materials library to select specific reagents. There is also option to lock fields once a particular information is entered, and if you need to alter such locked fields, the system will ask you to enter a reason for the change. In that way, the audit trail will capture all the changes that has been done to your experiment. The system also allows you to have plates associated with your experiment. In this particular case, you can see a 96 well plate with well format. You can describe the concentration of reagents used, all the materials that are used, and the cell lines that are used in this particular screening. All the materials which are part of the materials table will be available in the link add from experiment, and you can select the various different materials that are available in the materials table. If you have structured data in CSV files, you can use a template by downloading the template from the table itself. Populate the table with data and upload the table. The system will ask you to map the columns, and clicking on import will extract that data from the CSV and put it into the structured table, which allows you to search on the information that is present in structured table immediately. Again, you can write up your results and conclusions in a text box as described before. Files can be dragged and dropped into your experiment, which is in this particular case organized under the files. You can have page images and you can annotate those images, which is very easy. Say for example, in this particular case, this is a light chain. You can use boxes, arrow marks, and text to annotate your images. Any annotation that you have done to the image will be captured under the tag. If you would like to hide all the annotations, it's very easy to do so. I have an immunofluorescence image here. And again, I can say that this is a cilium, which immediately adds that tag. And I can also enter in the description that I've used ARL13V as marker for the cilium. When you have a particular term that you would like to search on, you can copy that and search for that particular term in the quick find, which brings back all the experiments where that particular mark or the term has been used. You can drag and drop Excel file, PDF, or PowerPoint or Word documents as part of your experiment. You can also create new Word, Excel, PowerPoint files by using the Add Content option here. Clicking on the file itself, you can see a preview. If you would like to read the file in more detail and clarity, you can expand the file and you can read. If you would like to download, the option is available to you as well. Any edits that you do to the file will be captured in the audit trail. You can edit a file by clicking on Edit in Excel Desktop application or Excel Web. Just to show you quickly, I'm going to make some changes to this document. And when I close it, it will be automatically saved onto the cloud. If you'd like to track all the changes that you have done, you can click on the history of the document. And as you can see, the changes that you have done, you can compare with the previous version. In this particular case, you can see that I have highlighted a large section of the file. If you would like to revert it to the previous version, the option is available to you as well. Sequence files can be managed within the Signals notebook. We have TestLegend sequence viewer integrated into the Signals notebook, which allows you to see the vector map in this particular case and look at the sequence itself and all the annotations and features that you have described in your sequence. Let us have a look at how you can sign and close the experiment. There are a few options available to you. You can add reviewers and keep the experiment open. You can sign the experiment and keep it open. You can sign and add reviewers to the experiment. You can sign and add reviewer for a two-stage process. You can also sign it yourself and then close the experiment. I'm going to select a sign and add reviewer. You can select the reviewers that are available on the system. Type in a comment and tick the box that you understand that this is the electronic signature of this record and send it for review. When I send it for review to Paul, Paul will get a notification in the bell icon, which he can click on that particular notification, which take him to the experiment. He has the option to go through the experiment, make sure that everything is all right, and sign the experiment. I spoke about collaboration within the Signals notebook. This is through the comments section that is available throughout the notebook. Say for example, if you would like to comment on the entire experiment, you could click on this comments option and you can tag users on the system. Say for example, in this particular case, I'm going to tag Paul and I say that please go through this experiment. When I send that, Paul will get a notification in his bell icon, which he can click and go through that experiment that I have tagged him. Clicking on the Materials Smart folder will take you to the Materials library. In this particular system, there are various different materials libraries that are set up, whether it's antisense oligos, different antibodies, cell lines, the compound library, or various other libraries that are highly configurable and easy to set up on the system. I am now looking at a monoclonal antibody library. Clicking on the unique identifier of a particular monoclonal antibody will take you into the detailed description of that particular entity when it was registered. The fields that you see here are all highly configurable. The system allows you to link an entity with other entities that are present in the library. In this particular case, this antibody is expressed from this particular vector. It can also look at the various different coding DNA sequences or protein chain sequences that are registered in the system. Different batches of the antibody can be captured on the system as well. When you look at the containers, you will see all containers where samples of this antibody is stored. Clicking on a container will take you to the actual location of that particular antibody. You will see a container ID as well as a barcode that can be used for calling in this particular antibody as part of the experiment that we have already seen. We can also print barcodes from the system. There are various different options for you to print the barcode as it is suitable for you in the lab. You can also see the amount that is available. There is option to update the amount from the inventory itself. You can also request this particular container by sending the request option from this interface. You can check in and check out this particular container. Say for example, if you want to move it from a storage freezer to a lab freezer, that is possible so that the location of the antibody can be tracked within the system. Synthesized compounds can be directly registered from the experiment into the compounds library. Again, a lot of information that is captured as part of that registration. Signals Notebook comes with the natural monomer libraries out of the box. But if you would like to create your own custom libraries, you have the option to create individual monomers or bulk upload custom libraries into the monomer library. Once you have the monomer libraries created and activated on the system, you can use a Helm editor, which is embedded in the ChemDraw element for creating and registering complex macromolecules. The natural monomer libraries come out of the box. So if you were to create a peptide sequence, you can click on the monomers for the sequence that you require, and that will be drawn into the ChemDraw canvas. If you are creating nucleic acids, the sugar, the base, and the phosphate monomers are available out of the box. If you have non-natural monomers registered, in this particular case, a thiophosphate, that can be used to create your sequence. You can copy-paste Helm notation into the text box, which will draw the sequence into the ChemDraw canvas. Recently, we have enabled Bill notation in ChemDraw, which is a simpler, easy to read alternative for Helm notation for describing peptides. You can copy-paste the Bill notation and it will draw the sequence into the ChemDraw canvas. By using this capability, you can create and register complex macromolecules into the materials library, which is part of the Signals Notebook. I have a couple of examples in my materials library. This is an antisense therapeutic, which is inclisiran. And as you can see, there are various modified residues throughout the sequence. You can also capture antibody drug conjugates and register them into the materials library. Let us now look at a chemistry experiment creation. First, I'm going to show you how easy it is to search for chemistry experiments that are already captured in the notebook. You can search by drawing structure by using filters. I have drawn DMAP to search for all the experiments where this particular reactant is used. You have the option to match the substructure or match similar structures or match full or exact, depending on what I would like to find. You can also use sticky filters to add more filters to your search. Say, for example, going to look at all the experiments created by Paul, and the system immediately search and return the hits for me. Let us look at a chemistry experiment now. A chemistry experiment has very similar structure like the biology experiment, apart from drawing the reaction itself using the chemdraw element. I have used a pre-drawn chemistry reaction, and notebook is very chemistry aware. There are a few tables which comes into the experiment when you insert the chemdraw element, and these are reactants, the reagents table, the product table, the solvents table, and the reaction conditions table. If for any reason you would like to hide these tables, it is very easy to do that using the hide stoichiometry table option in chemdraw element. There are few ways that you can draw reactions into the chemdraw canvas. You can either draw it using your familiar chemdraw interface. You can also use the ChemACX, which is Revit's curated approximately 35 million compounds that are available from various different vendors. You can also add reactants or reagents using the quick add option. Say, for example, if I were to add DMAP, searching for DMAP will bring up the materials library by using a sample that is stored in a container, or you can have from a hard-coded list within the system. Once you add your reactants and reagents, they will appear in your reactants and reagents table. As a chemist, you can adjust equivalence, define which is the limiting reagent, and you can adjust the mass that you would like as part of your reaction. The system will also show you safety information for the reagents that you have used in your reaction. To save time for the scientists and to work efficiently, Signals Notebook allows text snippets with placeholders. In this particular case, the text snippet is looking at the stoichiometry table. Any changes that you make in the stoichiometry table will be automatically updated in the text snippet. Say, for example, I'm going to change this to 20 milligram, and you can see that the amount will be automatically updated in the write-up section as well. You can also click on the placeholders and select the relevant information that is available from the stoichiometry table. Once you have created your products, you have a couple of options. You might want to send it for analysis to an analyst. For that, you can create samples using the sample option in your add content. I'm going to opt for a chemical sample, and the system will automatically pick up all the products that are created as part of the reaction. The samples will be populated into the samples table. Once the sample is created, the scientist can take a few actions on it. They can, if this is their final product, they can register the material in the materials library as a compound. They can also create tasks and send it to an analyst for analytical characterization. Once a task is created, the list of the tasks will appear in the tasks table. Every task gets a unique identifier, and the table also shows the request link, which is the experiment itself, and the sample ID is populated into the table as well for quick reference. As an analyst, they could come into their dashboard and look at their tasks wizard and pick up tasks from there. As an analyst, they could also go to the Tasks Smart folder and look at the tasks that are assigned to them. It is very easy to filter based on the analyst's name. Paul can take a couple of actions on this task. He can select the task and create an experiment to carry out the task. Alternatively, he can click on the task ID itself and attach the results as part of the task itself. Once that is done, he can say that the task has been completed. And the status of the task changes to completed. When the scientist comes to the experiment and look at the task table, you can see that the task has been completed by looking at the status. And by clicking on the task itself, you can see the results of the task. The other parts of the experiments are very similar to what I described already for the biology experiment. Now I want to show you a couple of other things that you can do to an experiment. If you would like to share this experiment with your colleagues, you can click on the sharing option and add any colleague's name that you would like to share the experiment with. You can either give full control or read-only access or just write access. If you would like to copy an experiment, Notebook allows you to do a complete copy of the experiment or a copy partial. A copy partial will allow you to pick and choose the sections that you would like to copy to create the new experiment, as well as the attributes that you would like to assign to the experiment. It is also very easy to export the experiment as a PDF. You have the option to export the entire experiment or pick and choose sections that you would like to export. If you have files attached to your experiment, your PDF will have all the files associated with it as an attachment. What we have seen so far is how Signals Notebook can be accessed and how we can navigate through the system. We have also explored how experiments can be created both for chemistry and biology. We have looked at the asset management as part of the materials library within the notebook. We have also looked at how experiments can be sent for review to a team leader or a group leader for approval. Data captured in the notebook can be exported and shared with your colleagues and collaborators. And we have also seen how audit trail is captured and how we can explore the history of your experiment. So the value that you get out of having a Signals Notebook is that everybody in your organization makes decisions from a single source of truth. That allows you to have consistency, traceability, and visibility of all the data that is captured in the notebook. This allows less oversight and more insight into the research that you carry out in your organization. If you would like to know more about the Signals Notebook, please visit our website at www.revitisignals.com. I take this opportunity to thank you for your time, and I'm ready to take any questions you may have.
I can see that we have a few questions, so if you are willing to take them now, that would be great.
Awesome, fire away. Thank you.
Yeah. First question here. Are there any data processing and analytics capabilities within Signals Notebook?
Yes, indeed. So Signals platform, which we call as Signals Research Suite, is an end-to-end R&D workflow solution, and this provides data capture and collaboration, data processing, and data-driven analytics as well. Now, the Signals Notebook works as the central piece of capability to capture and organize the data for the make, test, and decide cycle. We have seamlessly integrated with the Spotfire. I think some of the listeners today might have heard of Spotfire or might have used it, using it now or in the past to analyze and visualize assay data, such as plate-based dose response curves or SPR assays, qPCR, to name a few. We can connect instruments and systems and services to communicate bidirectionally with Signals Notebook via Signals DLX, powered by our partner Cytara. We also have applications to manage in vivo studies. We have a specific application called SIMA, which is Signals Image Artist, to manage and analyze high content screening data with the phenologic AI built into it. The majority of actions that users take in Signals graphical user interface are available in our public API, which can be accessed through the the Swagger, which is available from the user interface, which is available to the administrators. So yeah, Signals is not just the notebook; it's the whole platform, which is far more capable for taking your data through that whole discovery cycle. Thank you.
Oh, yeah, okay. Thank you for the answer, Nimesh. I have one more for you. Can you explain how software updates are done? Sorry, on the tenant and how to find information about new functionalities incorporated into the product.
Of course, yes. So Signals Notebook is a SaaS solution, and all updates will be deployed automatically to all customers. Users will get an in-app notification about the upcoming and scheduled updates. Usually, the updates are done in a time when the users are not actively using the product. For Signals Notebook Standard Edition, there are currently roughly 10 updates per year. That is around one update every 6 weeks. Smaller updates will be readily available automatically to the users. Bigger updates addressing new features may require an activation and configuration process performed by the system administrator in your organization. Updates for validated environment, for example, work through a different staged process because it's a slightly complicated and different process. We need to be a bit careful how we update that. List of new features, say for example, the latest capabilities added or the past capabilities which are added, can be accessed from the application after logging into the system. Administrators of the system are recommended to subscribe to the channel within our support news site, which contains more information about releases and other pertinent product information delivered through an email, so that they don't have to search for the link where that information is available. So if they subscribe, they will get an email with all that information.
Okay, thank you. Good to know. One last question for you before I leave you. How does Signal Notebook support collaboration with CROs? I think that's contract research organizations.
That is correct. Yes, yeah. So we have a dedicated application for CRO workflows, which we call Signals Synergy. And this was launched pretty recently in the last few months. What Signals Synergy enables you to do is to seamlessly integrate with the Signals Notebook and the wider Signals Research Suite to offer a secure slice of your tenant for interacting with each CROs through a collaboration portal. And this provides a fit for purpose workflows for sponsor, which is you as an organization, and for the CRO to exchange data. Now within Synergy, the sponsor users can request work, example, analytical requests or assays or studies, from external contract partners, which may be CROs or CDMOs, for example, using work orders. Now, within work orders, sponsor users can configure tables. These are, structured tables to selectively share samples or material information and specify their request. Say, for example, if it's a compound synthesis, you can specify that this is the amount that I would like to have, or certain assays to be carried out. These are the assays I would like you to do on these compounds, for example. Now, work orders can also include various files, such as protocols to be followed, or manuscripts which has published similar data in the past, something like that. Once the CRO users complete the work order, the CRO users deliver their results in associated experiments right within the Signals platform. So this is seamlessly, the same platform. You don't have to jump into another platform. Results can be delivered within the experiment via a variety of flexible formats such as, Excel file, CSV, or other file upload, or data entry into templated structured table itself. Alternatively, Signal Synergy offers a unique solution for contractor data exchange. Rather than requiring the CRO to manually copy and paste their data into a requested structured data format, the data exchange performs this transformation for you. Through a repository of templates, CRO and assay-specific report templates and associated parsing maps, unstructured and multi-tabular Excel-based reports generated by the CRO can be automatically transformed into structured data tables containing the report, metadata, raw data, and analytics endpoint for consumption into the sponsor's data lake. So I've spoken a lot about the Signal Synergy because we are quite passionate about this new product, which is going to transform how sponsors are going to interact with their contract research partners. Yeah, thank you.